The topic for present video is impurities in pharmaceuticals, their sources, and the related guidelines. I welcome all the viewers to the present video, and I hope after watching this video you will be able to answer the questions like what are the type of impurities, how these impurities are generated or formed, or from where the impurity come into the pharmaceuticals. You will also know the specific guidelines for each type of the impurities. This will be a very informative and short video. So be till last with the video to have the good understanding about the topic. See, impurities are the materials which are not the part of API, which is not the part of formulation, and these are not the excipients. And many types of the impurities are there in the drug substances and the drug products. Have you thought that can we make the drug or can we make the drug product without any impurity? So is that possible? So the answer is no, that is not possible, because whenever you are making the drug substance or you are making the formulation, some impurities will come from the APIs, from the excipients, or from the degradation. Many of the APIs or drug substances are synthetic in nature, and because of that organic synthesis, the impurities come into the final drug substance. Impurities are not the API, and these are not the part of drug product as excipient. Impurities may be solid, liquid, or gas in nature, and these may be organic or inorganic. The major sources of impurities are raw materials. The raw material may be excipient, raw material may be API. The impurities may get generated into the API and formulation because of the API degradation or API and excipient interaction. Then degradation and leaching from the container and closures. That is also the source of impurity. Then impurities are generally classified into various types. Like the impurities may come from the process, may be formed during the storage as degradant, contaminants, and may be from the packaging components. In some of the liquid formulations, leaching may happen from the container. So those type of impurities get introduced. The degradation may be due to temperature, light, moisture, or oxygen, and the drug excipient interaction. So these are some of the basic sources. Now we will see the types of impurities. First, see the organic impurities. These are organic in nature, and these may come from the synthesis or manufacturing process. The impurities may be starting materials. Generally, the key starting materials used for API synthesis get introduced from the raw material, and these, if are not removed, then they will be present in the API and also will be present in the formulation. So that is one of the source of impurity. Starting materials, then intermediates, if these are not purified or removed, then traces amount of the intermediates will be there in the API. Reaction byproducts, sometime in the organic synthesis of API, reaction byproducts will be there, and also the degradation products. Degradants may be formed during the processing or may be formed during the storage period. Many of the time, the degradants are also the active metabolites of the API. So, metabolite of the drug or metabolite is also a degradant impurity. Then related substances. So these are the impurities which have relevant structure or structural similarity with the basic API or basic drug substance chemical structure. Genotoxic impurities, these are genotoxic and carcinogenic in nature. These may also be present. Then nitrosamine impurities. Then NDSRIs. These are nitrosamine drug substance related impurities. So these impurities I have classified into organic impurities. See, the classification may be different in different literature, but the basic understanding will be same. Then inorganic impurities, residual impurities, and isomeric impurities will be there. Inorganic impurities are mainly the heavy metal type of impurities like arsenic, pulver, and lead. So these are heavy metal impurities. Generally, the residual metals or salts may come from the raw materials or reagents or from the manufacturing process equipments. So these are also known as the metal impurities. Then residual solvents. So residual solvents like isopropyl alcohol, methanol, toluene, benzene, and many others. These solvents may be used in the manufacturing process, and in drying they are not completely removed. So these come as residual solvent impurities. There are some classes of the residual solvents based on the toxicity. Then isomeric impurities. So stereoisomers or potential isomers that can arise from the API synthesis will come as impurity. So whenever you are dealing with the isomeric type of API which have isomers or enantiomers, that time be aware about these isomer impurities. Then chiral and polymorphic impurities. So chiral impurities are enantiomeric impurities when the drug is chiral in nature. That time chiral impurity is required to be tested. Polymorphic form impurities. See, different crystal forms of the same API may be present or may be formed during the crystallization and purification and also under the storage. So different form of the APIs may be present in the drug substance, and also these API polymorphic form may be present in the drug product. So polymorphic form impurities are required to be tested. Along with these, the API and the finished product contains water content. The water may be bound water to the molecule or may be unbound water. Then LOD, that is loss on drying. Whatever the solvents are present into the drug substance and the product which get vaporized after drying, those are called as loss on drying type of materials or loss on drying type of impurities. The loss on drying may involve solvent and water. Water may be used in the manufacturing process, so that test is required to be done. Or if the API or formulation is hygroscopic in nature, and if on storage or standing it absorbs the moisture from the environment, that time it will increase the LOD and the water content. Presence of the moisture can lead to degradation and may also have impact onto the stability of the API. That's why testing the water content is very much important. Also, water content increases the chances of microbial growth. So, water content is required to be tested. Now coming to the guidelines for the impurities. See, there are many of the guidelines for impurities. Mainly I am covering here the ICH guidelines, USFDA guidelines, and some of the EMA guidelines. So ICH Q3A is the guideline for impurities in new drug substances. ICHQ 3B is the guideline for impurities in new drug products. Then there is USP chapter 476 for control of organic impurities in drug substances and drug products. ICHQ3C is the guideline which deals with the residual solvent type of impurities. And if you see the USP chapter, there is a chapter number 467 for residual solvents. ICHQ3D, that is the guideline for elemental impurities. Then in USP there are two chapters, 232 for elemental impurities limits and 233 for elemental impurities procedures. For some of the impurity types and for making the decision for impurities, ICSQ6A is the guideline for specifications, test procedures and acceptance criteria for new drug substances and new drug products. And mainly these are related to the chemical substances. Then you should see the individual monograph for the product and for the API. Whether it is USP, EP or BP type of pharmacopoeia or IP, that time individual monograph will give you the complete understanding about the impurities. Then chapter number 1086 is for impurities in drug substances and drug products. ICH M7 deals with the genotoxic and DNA reactive or mutagenic type of impurities. So ICH M7 is assessment and control of DNA reactive mutagenic impurities in pharmaceuticals to limit the potential carcinogenic risk. Control of nitrosamine impurities in human drugs, USFDA guidance for industry is there for the nitrosamine type of impurities. Then European Medicine Agency has some guidelines for nitrosamine impurities. Then coming to the USP General chapter, 1469 deals with nitrosamine impurities. Some guidelines are there for testing the metabolite as a impurity. So that is safety testing of drug metabolites, a guideline from US FDA. So these are some of the guidelines I have enlisted here. Other guidelines are also available. If you go through those guidelines, you will get the complete understanding about the impurities. So this was the video regarding impurities types in the pharmaceuticals, their sources and the related guidelines. Impurity and their limits, their sources are required to be known to the pharma professionals. Impurity is a topic of great interest in the pharma interviews. Whether the interview is for R&D, QA, QC, production, regulatory affairs or clinicals, this is the topic of great interest by the interviewers. If you are able to give the answers, then it will increase the chance for getting selected. I hope you might have got good understanding out of this video. Thank you for watching the video and I request you to subscribe Pharma Learning in Depth channel and share these videos with your friends and colleagues. Thank you.